Hi everyone, welcome to Enlightened Biology. Today we are going to look at the next topic, microsporogenesis. Genesis means formation, so it is a formation of microspores. How does the microspores form? From the microspore mother cell, which is also referred as pollen mother cell, okay, by meiosis. Is that clear? So it is a process of formation of microspores from the pollen mother cells by the process meiosis. So where these microspore mother cells are present? Within the microsporangium. So where is this microsporangium? Inside the anther. So inside the bilobed dithecous anther, there are 4 microsporangium which are surrounded by tapetal cell. Within those tapetal cells, there is sporogenous tissue, and the sporogenous tissues are made up of potential pollen mother cell. Why do we call it as potential pollen mother cell? Because they have the capability to undergo meiosis. Each and every pollen mother cell has the capability to undergo meiosis to form pollen grains. Okay, now what happens to them once the microsporogenesis starts? Every pollen mother cell, that is every microspore mother cell, is surrounded by a callose layer. Remember that well, it's very important. Every pollen mother cell is surrounded by a callose layer. What is the ploidy nature of that pollen mother cell? It is 2n. So pollen mother cell, in short known as PMC. It can be also referred as MMC, stands for microspore mother cell. Okay, MMC stands for microspore mother cell, PMC stands for pollen mother cell. Both are same, both are present. Both the names refer to the same cell. Okay, and what is the ploidy nature? Diploid. How many nuclei are present? Just one nuclei is present. Okay, this is a pollen mother cell. A pollen mother cell is a meiocyte. What is meant by the word meiocyte? Meiocyte means a cell capable of undergoing meiosis. Cyte means cell. Meo refers to the process meiosis. So pollen mother cell is a meiocyte. So normally in meiosis there are 2 stages, meiosis 1 and meiosis 2. In meiosis 1, the cell divides into 2 cells and the chromosome numbers are going to be halved. Okay, and in meiosis 2, further division happen. Finally 4 cells will be obtained. All the 4 cells are haploid in nature. So you could see that this pollen mother cell is undergoing meiosis 1, forming 2 cells, and these 2 cells are haploid in nature, and these cells are referred as microspore dyad. You can see that 2 cells are attached with each other and surrounded by a callose layer here, and they undergo the next process, meiosis 2, forming a microspore tetrad. First forms dyad, then tetrad. And why it is a tetrad or dyad? Why they are called so? Because all the 4 daughter cells formed by the process are held together by callose. They are not separate, they are always together. So now each cell has to develop separately. How that is going to happen? The chemical which is attaching them, that is the callose which is holding them together, should be dissolved with the help of the enzyme callase. So that is the function of tapetum, isn't it? Producing callase enzyme. So that enzyme degrades the callose layer. All the 4 microspores which are in the form of tetrad now comes out individually. So let us have a overall look again. Pollen mother cell undergoes meiosis 1, forming microspore dyad, undergoes meiosis 2, forms microspore tetrad, which are haploid in nature. All the 4 daughter cells are attached together by callose, and the callose is dissolved by callase enzyme. So we get individual microspores. All of them are haploid in nature. Okay. Now each one of this microspore will develop into a pollen grain. How this pollen grain development occur? Microspore will undergo expansion and vacuolation. Vacuolation refers to vacuole formation, expansion refers to the size increase. So here you could see that the microspore, which is haploid, has increased in size and vacuoles have formed here. Nucleus is pushed towards one side due to the formation of vacuoles. This cell undergoes unequal mitosis. So you could see the asymmetric mitotic spindle over here indicating the unequal mitosis happening. Then that leads to the formation of a bigger cell and a smaller cell. So the bigger cell is known as vegetative cell or tube cell. The smaller cell is known as generative cell. So initially the generative cell is attached to the vegetative cell by means of callose layer. Soon the callose layer will be dissolved by callase enzyme. Finally the callose will float within the cytoplasm of the Finally the generative cell will float within the vegetative cell cytoplasm. So this is the final structure we are going to get. Let us look at the entire structure one time. Okay, entire process we are going to have a revision now. Microspore is a haploid cell which undergo expansion and vacuolation. So vacuoles formed here, the size has increased, and then it undergoes unequal mitosis because the nucleus is pushed towards the one side of the cell. So there is an asymmetric mitotic spindle formed here indicating the mitosis process. Then there is a development of pollen wall. Exine and intine are going to develop. So as a result of unequal mitosis, we got a bigger cell and a smaller cell. The bigger cell is a vegetative cell, which is also known as tube cell. The smaller cell is generative cell. Okay, initially the generative cell attached to the vegetative cell by means of callose layer. Then the callose layer was dissolved by callase enzyme, and you could see in the last diagram the generative cell floats in the cytoplasm of the vegetative cell. This is what happened. Okay, so this what we see here, pollen grain, is a male gametophyte, but it is immature with two cells. Okay, two cell, normally in many of the angiosperm the pollen grains are released at the two-celled condition. Okay, there is going to be one vegetative cell and one generative cell. Okay, now we are going to look at the structure of the pollen grain. So here is the pollen grain structure drawn in detail. You could see the layers here. So this is exine, the outer layer, which is darkened and shaded. Intine is the inner layer, and the bigger cell is the vegetative cell. Within vegetative cell there is generative cell. This is the nucleus of the vegetative cell. Okay, the vegetative cell has another name, tube cell. Hence this nucleus is also referred as tube nucleus. Okay, the exine and intine together referred as sporoderm. So what is this exine? The outer layer, and it is not a continuous layer. There are gaps. So the gaps are referred as germ pore. Okay, so this exine is made up of highly resistant chemical known as sporopollenin. The sporopollenin is formed from the Ubish bodies which are produced by tapetum. This is a highly resistant chemical. It is resistant to high temperature, actions of acids, alkali. There is no enzyme which can digest sporopollenin is found yet. Okay, and because of this nature it can form fossils. So it is a thick layer. It is uneven because it is discontinuous. You can see the gap is present over here. It is a germ pore. So the size of the pollen grain is around 25 to 50 micrometer in diameter. So coming to the next one, the intine. The intine is the inner layer of the pollen grain. It is made up of pectin and cellulose. So it is normally referred as pectocellulose. It is a thin uniform layer, and during pollen germination generally through the germ pore. So you could see the germ pore over here. Through this germ pore, in this area actually exine is absent, so intine is going to come out and grow as a pollen tube. So intine is actually covering the vegetative cell. So along with the intine, the vegetative cell is going to come out and grow as a pollen tube. This is the basic thing over here. So the next thing we have to look at is the difference between the vegetative cell and generative cell. Okay, so here is the difference. You could look at that. Vegetative cell is also known as tube cell. It is bigger in size compared to the generative cell. The generative cell is comparatively smaller. Why the vegetative cell is bigger? Due to the presence of abundant food reserve for the pollen germination, and generative cell have no such food reserve either. And this vegetative cell is larger cell with irregularly shaped nucleus. Generative cell is spindle shaped and it's smaller. It is going to float inside the cytoplasm of the vegetative cell or tube cell. So vegetative cell grows as pollen tube during pollen germination. Generative cell forms two male gametes on mitosis. Okay, so how this occurs? Let us have a overall view here. So here is the overall process. You could see what actually happens. The pollen mother cell undergoes meiosis, form microspore tetrad. Then the tetrad layer, covering layer, that is callus, dissolves, forming microspores. Each microspore undergo developmental process to form pollen grain. The pollen grain undergo mitosis to form male gametes. So say for example you are starting with one pollen grain. There is one pollen mother cell. One meiosis is going to happen. That results in the formation of one microspore tetrad. And this tetrad, 4 cells are together in the tetrad. That is why it is called tetrad. 4 cells are together. So 4 microspores are formed from that. That will develop into 4 pollen grains. And this will undergo one mitosis process. Each one will undergo one mitosis process. So 4 into 1 mitosis. So there is 4 pollen grains, each of them undergoing one mitosis. So 4 mitosis totally happening over there, resulting in the formation of 8 male gametes. Each pollen grain will form 2 male gametes. So here are the some of the formula. You can note down them. So number of pollen mother cell is equal to number of microspore tetrad. You are starting with one pollen mother cell, you get one tetrad. If you are starting with 4 pollen mother cells, you are going to get 4 tetrad. How do we calculate the number of microspores? If you are starting with 1 pollen mother cells, you get 4 microspores. Hence, if you are starting with say 20 pollen mother cells, 20 into 4, how much it is? 20 into 4, 80. So you are going to get 80 microspores over there. How many pollen grains you are going to get? Each microspore will develop into one pollen grain. If it is pollen mother cell, each one will result in 4 microspores. Say you are starting with 100 pollen mother cell, you will end up with 400 pollen grains. The same way the other calculations can go on. So please consider this. Please note down all the formula so that it is useful for you to go through. Right? Hope the topic is clear. If there are any queries, please let me know through your comments. Thank you.